Hi students, I would like to continue the lesson that is chapter 4 Principles of Inheritance and Variation. Those portions left for pure science students, only pure zoology. Now the first one here we have to talk about that is cytoplasmic or extra chromosomal or extranuclear inheritance. Normally, most of the characters are controlled by genes or factors found in the chromosomes, and the chromosomes are found within the nucleus. This is called chromosomal inheritance or nuclear inheritance. Nuclear chromosomal inheritance. Genes in the chromosomes of the nucleus transmit the characters. But in some instances, we have certain characters are controlled by genes, and these genes are present not within the nucleus but outside the nucleus, not in the chromosomes, that is outside the nucleus in some other components, particularly cell organs. So, those characters which are controlled by just certain genes, and those genes are present, that is outside the nucleus in the chromosomes or in the DNA found in some organelles of the cytoplasm. Such inheritance, which is normally occurring through the cytoplasmic organelles, is called cytoplasmic inheritance. That is not nuclear inheritance. It is called cytoplasmic inheritance or extrachromosomal, other than the chromosomes, or extranuclear inheritance, other than the nucleus. So either cytoplasmic inheritance or extrachromosomal inheritance, or we have extranuclear inheritance that we have to study here. So certain characters are controlled by, see that one, non-nuclear genomes. Non-nuclear genomes. I mentioned already they are found outside the nucleus. The genomes are found outside the nucleus in some cell organelles of the cytoplasm. For example, chloroplast, mitochondria, or we can say some infective agents or in plasmids. Mostly we are talking about inheritance through the chloroplast and mitochondria. Okay, as these organelles are found in the cytoplasm and transmitted through the factors of cytoplasm, the inheritance is also called cytoplasmic inheritance. Now, these characters do not reveal, do not show the Mendelian pattern of inheritance. Here, the inheritance pattern is different. We are not getting the same ratio as in the case of Mendel's ratio. So, these characters do not reveal there is a Mendelian pattern of inheritance. Now, normally, you know that one. The inheritance of extra chromosomal, just actually genes, are found to exhibit what is called maternal influence. That means through the egg. The maternal influence means through the female parent. What's the reason for that maternal influence? Now, the maternal effect is more in cytoplasmic inheritance. It is due to asymmetric contribution of the female parent to the development of zygote. Now, if we are taking what is called fertilization process, now this is the egg having large cytoplasm, then we have the sperm. The sperm contains very little cytoplasm. The major contribution for the development of zygote is coming from what is called actually the cytoplasm of the egg. That's why I say asymmetric contribution. That is, the sperm just contributes very little cytoplasm, whereas the egg contributes more egg cytoplasm or more cytoplasm. See, unequal just a contribution of cytoplasm of both egg and sperm, hence called asymmetric contribution, particularly by the female parent. It provides more cytoplasm to the zygote than what we got the sperm. So normally what is happening, although both male and female parent contribute what is called the chromosomal genes, those found in the nucleus, equally. Okay, they contribute equally in terms of chromosomal genes. Suppose they have 23 pairs of chromosomes, both the sperm and egg, they contribute equally the number of genes present in these 23 pairs of chromosomes of both. So both male and female parent contribute equally in terms of chromosomal genes. But you see that one, The that is a parent, contribute the zygote's initial cytoplasm, the first cytoplasm, and cell organelles. This is because the sperm contains only very little amount of cytoplasm. So although both male and female parent contribute equally in terms of what we have the chromosomal genes, now the female parent contributes more to the zygote during the development as the sperm contains very little cytoplasm. Okay, that's what's called asymmetric distribution. Asymmetric distribution of the female parent. That means female parent or the egg provides more cytoplasm than the male parent, that is a sperm, because the sperm contains very little cytoplasm. Suppose if you have the hereditary units present in the cytoplasm, these units are transmitted to the offspring through the egg, because the egg only contains a cytoplasm having the cell organelles like mitochondria. So, if hereditary units are present in what we have in the cytoplasm, they are transmitted to the offspring mainly through the egg, not through the sperm. Okay, so that the offspring exhibit maternal effect. They have more maternal effect than what we have actually, that is the male parental effect. Okay, that's about what we have, something related to the cytoplasmic inheritance. I mentioned already, if there are hereditary units in the cytoplasm and these units are transmitted to the offspring only through the egg, so that what's happening, the offsprings exhibit maternal effect. The maternal effect is more than what is called the paternal effect. Now, the cytoplasmic extranuclear genes have different, that is characteristic pattern of inheritance, which do not resemble the genes found in chromosomes, which do not resemble the genes of nuclear chromosomes, we can say, because they're found in the nucleus of the, actually found in the nucleus, there's a chromosomes found in the nucleus. So these genes, the extranuclear genes, do not resemble what we have, the genes of the chromosomes, genes of the nuclear chromosomes. That's why it's known as extrachromosomal or extranuclear or cytoplasmic inheritance, and exhibit the maternal influence. The maternal influence is more when compared to what's called the paternal influence from the sperm. Now, what is happening in extranuclear inheritance? Both male and female contribute the nuclear genes equally, but do not make equal contribution of the cytoplasmic genes or extrachromosomal genes. Okay, they contribute equally the genes present in the chromosomes. That's called nuclear genes or nuclear chromosomal genes. But they do not make equal contribution of what we call the cytoplasmic genes, or we have extranuclear genes, or we have extra, that is chromosomal genes, those genes found in the cell organelles. Hence, there is a difference. Hence, normally what will happen, these processes can result in difference or non-Mendelian results. We are not getting the result as in the case of Mendel's principles. We are getting a result different from that of the Mendel's, what is called the results of 3:1, etc. So that's about one. And two specific examples given in the book. So cytoplasmic inheritance can be studied through shell coiling in the case of one snail, Limnaea trigra. Another one, the kappa particles of what is called Paramecium aurelia. The cytoplasmic inheritance in animals can be studied with reference to two examples. One, shell coiling in Limnaea perigra, that is nothing but a kind of snail, a freshwater snail. Another one, the kappa particles in Paramecium aurelia, that is a protozoa. Now, in the case of Limnaea, the snail, we have two types of coiling. One, the dextral coiling, another one, sinistral coiling. Dextral coiling occurs towards the right side in clockwise direction, and sinistral coiling occurs in what is called anti-clockwise direction. The dextral coiling is due to what's called the dominant gene, and sinistral coiling is due to the mutant gene. Suppose there is a snail which just donates egg, that is having the dominant gene, is fertilized by a sperm having a recessive gene for sinistral coiling. The result will be dextral coil. Egg from dextral coiling snail, sperm from sinistral coiling snail resulted in F1 generation. We have all what's called dextral coiling because it is being contributed by what is called the female, the egg, the cytoplasm. Suppose the egg cytoplasm, or we have the egg from the sinistral coiling, is fertilized by a sperm from the dextral coiling. We have received individuals with the sinistral coiling. Though the genes are present in what we have, the nuclear chromosomes, normally it is a cytoplasm which decides what type of individual develops into. So egg from dextral coiling, sperm from sinistral coiling, the result will be dextral coiling snail. Then egg from sinistral coiling and then sperm from dextral coiling, the result will be sinistral coiling. So this is a maternal influence that results in the development of what is called an individual. Accordingly, we have either dextral coiling, what is called the clockwise coiling, or sinistral coiling in the form of what is called anti-clockwise coiling. Similarly, in the case of kappa particles present in the cytoplasm of Paramecium adelia, those Paramecium with the kappa particles, they are called as killer Paramecium, and others without these kappa particles are called sensitive Paramecium. Here too, when the cytoplasm having the particles transmitted to what is called another Paramecium during conjugation, they become what is called actually individuals with the kappa particles. So we don't have any explanation in the book. Anyway, this is the one, just only I am telling the example. So egg from dextral coiling, then sperm from sinistral coiling, the product is dextral coiling. Then egg from sinistral coiling and then sperm from dextral coiling, the product is sinistral coiling. So here we have the maternal influence. The egg cytoplasm carrying the factors responsible for the expression of the character. Accordingly only we have, that is the development of shell coiling in the case of Linnaea. Different methods are adapted to improve the quality of human race in future generations. And for that we have different methods. The different methods adapted are eugenics, euphenics, and then euthenics. Eugenics, then we have euphenics, and then euthenics. Now what is eugenics? Application of the laws of genetics. Application of the laws of genetics for the betterment of human race, for the improvement of human race in future generation is called eugenics. So the application of laws of genetics for the improvement of human race is called what is known as eugenics. This concept, just actually eugenics, means well born, making the individuals born without any what is called defective characters in future generation. This concept, the term eugenics, was first coined by, that is Sir Francis Galton in 1885. Okay, so for the betterment of human race, we have to increase the population of what is called outstanding people. There are two ways. Either you have to increase the population of outstanding people, or to decrease what we have the abnormal or defective people by adapting the principles of eugenics. These are the two ways, either to positive way or to negative way. So, for the betterment of future generation, it is normally necessary to increase the population of, that is actually outstanding people, that is people with the desirable traits, or to decrease the population of abnormal or defective people. And this concept is called what is known as eugenics, either to increase or to decrease by applying principles of what is called eugenics. I mentioned already, so we have to increase what is called just good quality people, and we can say outstanding people, or to decrease the defective people. So there are two methods of eugenics. One method is called constructive method. One method is called constructive method. Another method is called restrictive method. Constructive method is otherwise called positive eugenics. The restrictive method is otherwise called negative eugenics. So in positive eugenics, we are increasing the number of that is outstanding people in a population. In a negative eugenics, we are decreasing the number of what's called the defective people or abnormal people by adopting what is called the principles of eugenics. Okay, now we have to go what is positive eugenics and what are the methods adopted here. Now the positive approach, the constructive method of eugenics, that is positive eugenics. Now this positive eugenics attempts to increase consistently the better or desirable what is called the germplasm. Okay, germplasm. The desirable germplasm is increased. And to preserve the best germplasm in the society. That is what is called actually positive eugenics. Now the desirable traits can be increased by adopting the following measures. Okay, either that is to increase the individuals with the desirable traits, or you can also the negative ones suppressing I mentioned about. Now what are the methods adopted, the measures here? Now you see that one, the positive eugenics, promoting the individuals with better quality. Then what we have, just interval between the subsequent pregnancies and between the pregnancies. Then early marriages, with the desirable traits to be increased. And the individuals with what is called actually more age. That means late marriages should be normally avoided because late marriages resulted in the development of certain syndromes, mainly for example the Down syndrome. Now see what are the measures adopted here in positive eugenics. Now early marriage of those having desirable traits. This is number one, early marriage of those having a good quality or those having desirable traits, early marriage. Then subsidizing the fee, so providing monetary benefit or anything else to them, and also establishing the sperm banks and the egg banks to just normally preserve the precious germplasm. The precious, more important germplasm with the desirable traits have to be just done through the sperm banks and egg banks. Then we have to educate the people about the principles of the basic principles of genetics and eugenics. Then only they can adopt the measures. Then improvement of environmental conditions, better what is called life. The better life, providing better life either through monetary or through what is called actually providing other conditions. We can just promote the individuals to produce more number of offsprings with the desirable traits. Then promotion of genetic research. So we have to proceed the genetic research to find out the better things, how to increase the population with the desirable traits, or how to increase the population of outstanding people. So for that we have to make genetic research to find out the better way to increase the number of individuals. So that's about positive eugenics. The next one, negative eugenics. We eliminate the individuals with the defective germplasm by adopting the following measures, different measures. For example, birth control methods, isolation, otherwise we can say separation of individuals with the defective characters, sterilization, what I mentioned, the birth control, okay, etc. Let's see one by one. What is negative eugenics? How can you just adopt this method to prevent what we have, the individuals with the defective traits? Now, negative eugenics attempts to eliminate, its importance is normally to eliminate defective germplasm. That's the main purpose, eliminate defective germplasm of the society. Those individuals having genetic disorders or any other defective characters will not be allowed to reproduce. They will be allowed to survive but will not be allowed to reproduce by adopting the following measures. 1. Sexual separation of the defectives. This is called isolation. Provide counseling to those having genetic disorders and just advising them not to have reproduction, to prevent reproduction. So that is called sexual separation of the defectives. Then sterilization of defectives, that's nothing but the birth control methods. Through birth control methods, one can prevent the spreading of the disease. So there is no possibility of transmission of the disease because of what is called actually sexual intercourse or mating. Then control of immigration, avoiding the individuals from other areas to enter because they're responsible for the introduction of certain genetic disorders. Then also regulation of marriages. So avoiding late marriages. Late marriages I mentioned already. So we have to have only the early marriages at the right time, avoiding the late marriages. So in late marriages, there is a possibility of getting what is called the syndromes in the newborn child. That's why that should be regulated. That's called the regulation of marriages. Now the second method for the betterment of human society, how to improve the human society, eugenics. Now once you have the genetic disorders, the way any disease to be treated immediately based on the symptoms. So the symptomatic treatment of genetic disorders, that is diseases of man, is called what is known as eugenics. So based on the symptom, that is called symptomatic treatment. This is what is called symptomatic treatment. Or genetic disorders or diseases, okay, of man is called eugenics. Otherwise called as medical engineering. Now, this term was coined by leader, actually Joshua Lederberg. Joshua Lederberg in 1960. So two persons, Joshua Lederberg and Esther Lederberg, they were responsible for just showing that one certain mutations are advantages. That was a concept developed by what is called Joshua Lederberg and Esther Lederberg. Now, this concept, that's eugenics, was coined by Joshua Lederberg in 1960. What is the meaning of eugenics? Normal appearing. That means without any disease. That's the meaning for that one. For just normal eugenics, well born. Now eugenics, normal appearing. That's about what we have. Actually, there is a few words about eugenics, and we have to go further. Now we have seen already that is what's called eugenics, the symptomatic treatment. So normally this method deals with the control of several inherited genetic diseases in humans, especially with reference to inborn error of metabolism. So inborn error of metabolism, you know that one just Garrod explained about the inborn error of metabolism. So the biochemical pathway is normally affected because of a mutation of a particular gene that results in a disorder right from the birth. That is called inborn. Error means in the genes, mutation. So we have one example, phenylketonuria. It is due to inborn error of metabolism according to Gerard. So that is called metabolic error due to metabolic blow. That is because of mutation in gene. So such diseases can be cured by using what is called the method, that is eugenics. Now the last method to improve the quality of human race is euthanasia. Unlike the following genetic principles, here we have to follow strictly the environmental management. So maintenance of environment is another method of improving the human, that is betterment. So it's a science of improvement of existing human race, not actually in the case of eugenics, we are getting better future generation. But this method is normally adapted to improve the quality of human race of the existing what is called population. So the science of improvement of existing human race, not the future generation, existing human race. By improving by improving the environmental conditions, that is called what is known as euthanasia. It can be achieved by adopting the following measures to provide better nutrition, better unpolluted ecological condition, there is a surrounding, better environment, then better education, and even better employment also. That is coming into this category, better employment. Then sufficient medical facilities to just avoid the death of the individuals and also to prevent the spread of the disease, so that we can maintain a better what is called population. So by improving the environmental conditions, one can maintain what is called the better that is population, and that is we can do that one for the improvement of human population. That is called euthanasia, improving the environment conditions for improving the human race. So with this, I concluded about that is the various aspects of eugenics, euphonics, and euthanasia, and also that is the last part of the chapter 4, principles of inheritance and variation. With that, I just concluded all the lessons for pure science students. So no more actually the lessons for you. It's your duty to study the lesson, go through the videos because I completed all the lessons. Okay. So thank you. Go through and just to get prepared well for the forthgoing examination. Thank you.